first session. We now will have a session on new diagnostic tools and treatment of child diseases. So the first speaker, when we are ready, is Dr. Guillaume Smits from ULB, and he will talk on clinical genomics and pediatrics.
First, I would thank the organizer to give us the opportunity to present our results here today. And so the work we present has been done by the four persons here, 2 PhD students, Dr. Belgian, pediatrician geneticist, and myself. And so what we have seen in the past couple of years is a shift of the pediatrics clinical genetics activity to clinical genomics activity. The clinical genetics was an activity where you had mainly you were seeing the patient and you had to cherry pick one gene according to the clinics of the patient. And you could test it by Sanger sequencing or by a cytogenetic single gene test. Now, we see patients where our best option is more to apply more genomic tests, where, for example, in cytogenetics is the CGH array, where we test for all the coding genes that are known, and for exomes. And our work is first to recognize the patient that would benefit from those genomics tests, and secondly is to interpret these very complex tests in an educated way through experience and literature, and to combine the clinics of those patients with the result of the test and trying to pinpoint which variants, which genetic variant could explain the phenotype of the patient. And if we believe this variant is the culprit, or several variants are the culprit, to be able to do a genetic counseling after that for the patient, for the family, for preconceptional clinics. And here, so one point we've seen is that now there is 4 big phenotype categories to help us to pinpoint a patient that would benefit from a genomic test. And two families could be one, as the neurology and psychoneurology, and you have a global developmental delay, epilepsy moderate or severe, and actual hypotonia that are quite frequent symptoms in the patient we see. In psychoneurology, you have the cognitive phenotypes, you have the behavioral phenotypes, and more seen in a teenager or adult, and so more seen with the parents or with the siblings, you can have the psychic phenotypes too. And you have also the classical anatomy phenotypes, so the malformations, and now it's clear that growth and mainly failure to grow, and failure to thrive is a good symptom to select the patient that would best benefit from a genomic test. I have put a red plus for symptoms that do need to be, where you need more, several symptoms to think it's a good, a genomic test would be a good indication, while some like global developmental delay or multiple congenital anomalies or moderate or severe intellectual deficiency or autism, for example, by themselves could justify a genomic test directly. You also have to pay attention compared to the classical clinical genetics activity we had, is such a tree in the past would have been considered not very informative. Now, in the genomics days, we have discovered that atypical presentation and incomplete penetrance is probably the norm of a genetic disease, except the very famous one, like cystic fibrosis, for delta F508, but in genomics, we are dealing mainly with, we have seen the appearance of risk factor genetic variants, and now research is based on oligogenic disorders, where you will have several variants that would explain the phenotype of your patient. And in this tree, where this patient had global developmental delay and was seen in our clinics, what was found is a deletion, so a CGHR was done, a deletion of NRX and one deletion of exon was found, and this patient has the deletion, but the father was an asymptomatic carrier, and it's very probable that those two patients, because they correspond to the genetic phenotype spectrum, were also carrier of this deletion. So atypical trees, atypical family history need also to be kept in mind when you are in clinical genomics clinics. So what are the tests that are available to us now? So the patient arrives in clinics, the first thing you do is that you look for a pathognomonic sign. If there is a pathognomonic sign, and if there is a low genetic heterogeneity, for example, if a clinical sign is very specific of one gene, one disease due to one gene, then you can go to the classical clinical genetics protocol, and you will go for a single gene or a small gene panel, for example. Like for, there are 5 or 10 genes. It would be cost effective. But for the big majority of the patient, the choice of one test will not be cost effective, and the clinician will be often wrong. And there are papers, for example, from Baylor, where they show that the doctors sequenced all the Noonan genes, and then it was a coffin series. And they sequenced all, they were sure that it was a Noonan. No. The atypical presentation is probably the norm. So it's better to work with agnostic tests and then to apply your clinical skills to the results of that test. So in first line we will do a CGH, and now a lot of children's hospitals are trying to develop in second line the exome sequencing, but here we have maybe 1 to sometimes very rarely 4 or 5 variants to interpret. Here the problem is that we will receive 20,000 variants or even more to filter and then to clinically interpret. So the time of medical interpretation is enormous, and there is still a lot of work to do here. In some centers, they will now apply whole genome to the patient. So how did we try to implement clinical genomics in our hospitals? First, we had to gather bioinformatics. You can't do it without bioinformatics, and so we created with our sister Flemish Free Sister University on one common campus at La Plaine, the Inter-University Institute of Bioinformatics and Research, and it's a joint venture between the engineers, the scientists, and the medical doctors for both universities. And this is really needed because the computing science will come from those two faculties, and the biology will come from this faculty for the patient. In science, you can have also those two. Second, you need to have the ethical approval. That was not an easy task, because you have two issues mainly at play. One is the incidentalome and the secondary variants. So because we are, for example, in exome sequencing all the genes, there is a risk to see variants that do not explain the phenotype of the patient, but that could be clinically actionable. And that's very a problematic issue, because there is no good solution. If you see one, do you, and it's actionable, do you return it automatically to the patient, or do you give the choice to the patient to say, no, I don't want to know? But how do you explain? You can explain for one disease, but how do you explain for 50 diseases or 100 diseases in half an hour or even an hour of genetic counseling? So there is a big debate in the world for the moment about the incidentalome, and there are a lot of literature. and the sharing issue. So the data sharing is very, very important in rare disorders because some patients are close to unique in the world. And so if you can't share the variants of your patient in database, in world databases, you can't maybe explain your patient because you will need 2, 3, 4, 5 patients that have the same variant or that have a variant, a strong variant in the same gene to be convinced that this variant explains the phenotype. First, for your patient you need it, but also to help you need to share. And that goes against the privacy issues, and that goes against the European law that is in discussion for the moment. So the Rare Disease Parent Association are really fighting back this law because it would impeach a lot of the sharing of genomic variants. Third, as I said, the atypical and incomplete penetrance force you to widen the genomic testing indication. So you have to do that carefully because you don't want to be swamped, because it takes a lot of time, but at the same time you don't want to miss a diagnosis with a patient because we have seen it's a very powerful test. Fourth, you have to empower the non-geneticist clinicians. You have to train them, you have to explain them how to what is a genomics test, how to prescribe it, how to recognize the patient that would benefit from it, and then to also teach them the limits of those tests. Those tests are very, very powerful, but they are not perfect. And fifth, we are there for the moment, so you have to prepare a way to study your results globally by bioinformatics to be able, and then electronic medical records, databases, local databases are very important. You have to improve your analysis and reproducibility. You have to feed back constructive conclusion to routine lab and clinicians, so feedback like that they can progress all the time too. And you have to create systems that will automatically share global with global and local databases. To diminish the risk, you should memorize this word. It's variant of unknown clinical significance. And this will help to avoid this. So what I will present here as data rapidly is the problem that we will encounter with the personal genomics issue. So while in research, Exome has been fantastic to discover a lot of rare disease and Mendelian disease and the etiologies and which gene was the culprit, and we have discovered a lot of things. For the patient, very often you will have one patient. And then when you will apply your exome, you want the exome to be clinical grade. And it will not be the same as research. And the two examples, I will show you two examples here. So exome in research. We had a chance that Dr. Villain and your team have collected a series of 6 cases of an ultra-rare phenotype, the Hartsfield Syndrome phenotype, where patients do present holoprosencephaly and ectrodactyly. Holoprosencephaly is anomaly of the medial line of the brain, and it can be major or with microforms, and ectrodactyly is the central rays of hands or feet that have anomalies, that are missing. So we had 6 cases, so we received money for 6 exomes. But we decided because we know that exomes are plagued with false positive, we decided to do a dual strategy. First, so that's a 4 patient from the most severe and the most minor form, and we thought because it's plagued by false positive exome, there was a risk that if we sequence 4 ultra-rare patients, we do not pinpoint the gene directly. And this was indeed the case. Strong variants were found in 260 genes. So if you want to go to 260 gene PubMed literature and to know which one is the culprit, and very often when people do it like that, they are wrong. So what we have done is that we combine the series strategy with a de novo strategy. A de novo strategy is to do, and it's very powerful in clinics. The problem is that you have to do 3 exomes, 2 for the parents that are asymptomatic, and 1 for the child that has the presentation. And Hartsfield Syndrome was always sporadic. So there was a good chance that it was a de novo mutation. And you can have 75 genes or 10 genes with stringent quality criteria. So that's possible, at least 10 genes, that's possible, but even if you go 15 minutes per gene per PubMed, time 10 for a medical doctor, that's a long time. Huh? So these two strategies allow us, when we do the combined analysis, even with a messy one, the only gene that was left was FGFR1, where we had strong mutants, de novo, and for the 4 patients. And biologically, it made a lot of sense. I do not have time today to describe it, but the hospital offered us open access, and so you can go and see it. So exomes are plagued by false positive variants, and so in for clinical grade exomes, we will have issues there. So we decided to work on the reproducibility of exomes. There are 2 types of reproducibility: the classical clinical biology issue, which is the analytical test production reproducibility, and then the bioinformatics reproducibility, which is very new in our field. And so for analytical improvement, mainly you have different strategies, and all those strategies try to get better coverage and better sequencing depth per gene. And so simply here, for example, this region is missing, and then you could get a better region. I don't have time to go in detail, but what we can hope is mainly in long term is better sequencing machine and to be able to have whole genomes. That would be the best strategy. Now on bioinformatics improvement. So here, this is what gets out of the sequencing machine. And you will have millions of those small reads, ACTG, that need to be realigned to the genome. And then you have several algorithms and software that do that. Then after that, you have to kind of correct your alignment. Then after that, you have to call your variant. That means compared to the genome of reference, you will see that sometimes the patient is heterozygous for a variant or homozygote for a variant that is not the one from the genome of reference. And that's all the bioinformatics pipeline, and a lot of labs in the world are using the GATK pipeline developed by the Broad Institute in Harvard. And we got money from the Brussels region to do an exome pipelines comparison. So we got money to harmonize the NGS pipelines in Brussels. And so you have the 3 academic centers and an exome company. And as you can see, we are all using close to the same tools, but not the same version, and some different tools here, and they are all highlighted, or some other strategies, single sample at a time or multi-sample. So you see in color that we, even in Brussels, we work closely, but not the same. And does these differences do create a difference for your patient? If I order an exome at UCL or at UZ Brussels or at ULB, will I get different results? This is a question that was not yet answered. So we use 6 exomes and we gave the sequencing files to the patient, so just what out of the machine, sorry, to the researchers, to partners, and they applied the bioinformatics pipeline to those same 6 sequencing files. And this is a representation of the results. And here you can see that for SNP, so single nucleotide polymorphism, we get an up to 10% discordance between 2 pipelines. And here for the indels, and we know indels, it's for example some 2 or 3 letters that are missing or 5 that are incorporated in the DNA, so very short, that we can see in sequencing. It is even worse, up to 25%, and this is because indel mainly are creating a problem with the alignment. So I do not have time to present you more and more detailed analysis, but I think that in conclusion we can I showed you that in clinics we encounter a pediatrics drift from clinical genetics to clinical genomics. And the tools are different, the way we perform our clinics is different, and we become more and more, we have to integrate the interpretation with the clinics. it really needs a multidisciplinary team. Bioinformatics is crucial. We need new coding for consent and for sharing. There is a need for analytical improvements, so the exomes are good, but they are not perfect. People say that on average it's 80 to 90% of the exomes that are well covered. So there is a need for bioinformatics reproducibility to arrive to an exome, clinical grade exome. Some centers are already there, you know, the one at the cutting edge, but in Belgium we are working on it. And we need also to adapt the reimbursement to bioinformatics needs, because a major part of our work now is in front of a computer, and its interpretation. The INAMI visit does not give any money for that. So clinically, what the take-home message would be that there is this balance between the four big categories of phenotypes to recognize a patient that could benefit from a genomic test. I here do not, I separated the patient that, for example, would have a very specific pulmonary disease for which we know 15 genes, for which there is a panel of genes. Now, for example, the fever, a patient, a clinician will see the fever, he knows that there are 8 genes, and he will ask for the panel. Here, the genomics, what I call genomics, is for the 20,000 genes, or for a lot of genes at least, not for 5 of them. There is, you have to remember that probably incomplete penetrance and atypical presentation is the norm, and that genetic counseling for those risk factor variants, where, for example, a patient has a risk factor variant, and you don't know which is the second hit, or the third hit, fourth hit. Your genetic counseling is very complex, and certainly in the prenatal setting. So these risk factor variants, for example, there are some structural variants, some deletions or duplication that are risk factor variants, are a real problem in prenatal setting. And 8 Belgian centers have recently published a paper in the European Journal of Clinical Genomics, where we tried to create a protocol of how to answer those the structural variants we found at CGHRA in a prenatal setting. And these risk factor variants are a real big issue. And now, for the moment, we are doing research on oligogenic disease. So I would like to show you, to present you the team that has done all the work today. And Dr. Villain, who had a series of Hartsfield patients, Professor Jean-Luc Bontempier, who is the director of the IB spread, and Bertrand and Claudio that have done bioinformatics analysis. I would like to thank Professor Casimir and Professor Lepage for the strong support to help us to build this clinical genomics clinics, and the Belgian Kids Fund, and all the funds that help us. Dr. Simonis, a bioinformatician who has helped us to start the pipeline and was key to the Hartsfield paper. The people from the ULB and VLB genomics platform, so now we have shared machines because they are very expensive, and we share expertise, and there are a lot of people in the 2 labs that do work there. And here are all the people that work on the harmonization of all Brussels and the analysis, and to develop new analysis tools, and from ULB, from VLB, from UCL, and from in Singapore. Thank you very much.
Thank you for this very interesting paper. I'm sure that there will be a lot of questions from the audience.
Thank you. When you identify a mutation in a gene, looking at a trio or a family, you then need to show that that gene is dysfunctional, or the function of the gene. So how do you propose to take that information and then show that that gene is actually a dysfunctional gene? Do you use cell lines?
So now it's why the sharing is so important. So we were discussing that with Nemig and Beto. The genetic proof is still the best and the most easy to obtain. So there was a discussion, how many patients with a strong variant in the same gene do you need to convince yourself that this is probably the gene, the culprit gene. So I didn't show you, but for the Hartsfield, we had 2 more patients. We sang a sequence, we found the FGFR1 mutation also, so it became quite difficult not to believe it with all the biology that was available. So that's the best proof you can, and this is why sharing is important. After that, or you have a lab that is expert in testing the gene you found, and he accepts to test it, or you have to develop tests, but that's impossible in a routine lab. So after that, some labs try to do quick animal models. And for example, one that has got a lot of attention is the zebrafish, because it's a vertebrate and it's easy to see. And so some labs, for example, in North Carolina, Professor Academies has developed a lab where they can test zebrafish mutants and see, for example, if it gives microcephaly, size problems, if there is an additional argument to believe that this variant is functional. And for example, also in epilepsies, Antwerp and Kai Leuven have worked together, and they have developed, and this is quite nice, electroencephalogram for zebrafish, and test of epilepsy for zebrafish where they record that it goes differently. So the cheapest would be to share the variants, to have databases, and to have other cases in the world. Otherwise, if you need really to convince yourself, you need to go very often to in vitro or animal tests and to collaborate with experts.